I'm very excited to have Agata Misiaszek here from EMBO Heidelberg, presenting us a human molecular machine and what does it do with cancer. Agata, the floor is yours.
Okay, thanks a lot. It's a pleasure to be here, and I hope you can still stay up with me, even though this is the last session, the talk of today, of this session. So I'm Agata Misiaszek, and I'm a PhD student at EMBO Heidelberg in the group of Christoph Miller. And I would like to take you back a little bit more to the basic research in biology. So let me start with something that, well, let me start with this moves. Okay. With something that is for molecular biologists, the big picture. So a cell, this is a big picture for what I'm working with. And what we are going to focus on is the nucleus, which contains DNA, so-called the code of life. But for the code of life to actually work, it needs to be read out. And it's read out by a special machinery, so-called RNA polymerase, which actually goes and transcribes it into a information carrier, which is RNA, which you might have heard of in the context of vaccines or of the last talk. And then this RNA can be read out by a different machinery called the ribosome. And the ribosome is a factory that makes proteins. Proteins make essentially everything in the cell. RNA polymerase is a protein complex, and you can find them all over the cell. Ribosome, though, is a special machinery which is only partially made of proteins, and in majority it is made of special type of RNA called ribosomal RNA or rRNA. And this ribosomal RNA is made by a special protein complex, special RNA polymerase called RNA polymerase I. And this is the focus of my PhD project. And in normal cell, this whole situation needs to be in a very precise balance, but sometimes our cells get a little bit evil and they transform into cancerous cells. So one of the main features of a cancerous cell is that it divides uncontrollably. And you can imagine that if a cell wants to divide very fast and make copies of itself, it needs to have a lot of the building block. So it needs to have a lot of the protein. And if you need to have a lot of protein, you need to have a lot of the factory. And to have a lot of the factory, you need to have a lot of the building block for the factory. So this whole process needs to be upregulated for cancer to progress. And that's why RNA polymerase I is actually a great cancer target, because if we can block this process at the very beginning, we can stop the cancer progression. But there's a different situation in which upregulation of this entire process is actually healthy. So if you want to develop from one cell to an entire human, you also need to divide the cells a lot. And in this case, if there's any issue to the RNA polymerase I, we observe disease such as Stretcher-Collins syndrome, which was showcased in this movie Wonder, which is a craniofacial developmental disorder. So you have issues with the development of the face, as was showcased in the movie. So far, this whole process has been studied mostly in yeast. So this is a unicellular single-cell fungus that is used for making beer, for example. And it's very easy and very accessible and cheap. But I think that I don't have to convince you too much that yeast are not exactly humans. Well, they don't suffer from cancer, they don't suffer from developmental disorders. And it is really important for us to look into the system in some more close situation to ourselves to be able to think about drugging it. And this has not been available due to technological hurdles so far. And with the advent of genetic engineering, which I'm using, the technology called CRISPR-Cas, CRISPR-Cas9, it's now possible. So what we do in my lab is we can target a specific part of the genome of the cell with this machine, with this molecular machine or molecular scissors, which are called Cas9. They can find precisely spot in the genome and introduce a cut. And our cells really don't like having their DNA cut, and they will do essentially anything in their power to repair this cut. So they will go in, and if we give them a little bit of additional DNA with a modification that we like, they will take this additional DNA and use it to repair the cut as a template, incorporating our additional part of the DNA kind of inside. And what we can get thanks to this is our protein complex of choice, so RNA polymerase I, with a nice shiny tail that we can then use to pull out this protein out of the cell and have it cleaned out. And then this sample is ready for my further investigation, which are structural investigations. And my technique of choice is cryoelectron microscopy or cryo-EM. So what we do is we take this pure protein in liquid and put it on this teeny tiny grid, which I wish I could just pass around in public to show you. So they're very tiny and they have super tiny holes inside, like a micron across. And we put very thin layer of this protein and freeze it super fast. And because we freeze it very fast, the ice crystals don't have time to form. And we have this amorphous layer, like liquid-like layer of ice, in which our proteins are embedded, hopefully in random orientations. So then when we put it into the electron microscope and image it, so this is one of the electron microscopes that we use. It's massive. It's essentially 3 meters tall. And we can image with this microscope, these proteins hopefully in random orientations with very low electron dose. And the image that you get is something like that, which probably doesn't look too convincing to people who are not used to looking at this. It also took me a while to start seeing anything in this gray image. But with a trained eye, or more likely some computer software, you can pick little images in here. So you can see, for example, here in each of the circles is an image of a protein. And then what we do is we crop out this little images and we can average them out and sort them together. So we can make similar groups, we can group similar pictures together, and we can get something like this, which already has a better noise signal to noise ratio. So you can start seeing some features in here. You can start seeing this Pac-Man kind of look that we are going for. So this is what we are going for. And then what we do is, because this is a projection, so it's a 2D image which encompasses the 3D information, and we can then take them, arrange them in 3D space with a lot of complicated computational algorithms, which I won't go into, and we obtain a 3D volume, something like this. So this is what I got out. And then afterwards, we can go in, and with our knowledge of the complex, we can start interpreting it. So we can start assigning different parts where they are inside of the complex. And then based off of what we can see in 3D, we can build in the atomic model, something like this. So here, by these ribbons, representing single atoms or single amino acids more, which are connected to each other. And we are creating this beautiful, to me at least, model of the protein complex, which illustrates what really is inside your cell, essentially. And you might be wondering why we went to so much effort to obtain this, and what do we do with a model like this? So the point of this entire exercise is that we can now look into great detail into every part of this model, and we can start answering questions. So first, let me take you to this part of the model. So this is where the RNA comes out of the whole complex in here. And if we zoom in, we can start seeing that our RNA in the model is twisting. It's forming like a double helix, almost like a DNA. And this is something that has never been seen before inside an RNA polymerase exit tunnel. And we were very excited to see this because it means that the tunnel is wide enough that this can actually fit. And we also think that this might help the polymerase go faster. And this is really important in human cells because the gene that it's reading, it's really long, so it really needs to go fast. So this is some adaptation that we hope that we can explain in human RNA polymerase 1. Another place where I would like to draw your attention to is this thing on the top, which we call stalk, like the mohawk kind of that the polymerase has on top. And if we zoom into this one in yeast, you can see that it's made of 2 pieces. So you can see this big blue piece and the small pink piece in here. So this is how it looks in yeast. And then once we look in the human, it turns out that there's only the blue bit. And this is a long-standing question that scientists who study RNA polymerases had, because we knew that yeast has this little pink piece, and we knew that we don't know if it exists in human, if there's something else. So only this year, which is I think pretty late, we can finally say that no, in fact, there isn't anything. Humans just have one less piece. And when we looked into the tree of life, this is the conserved feature. So most of the organisms have only 1 piece, and yeast is the outlier. So scientists many, many years ago managed to pick an outlier and start making assumptions about how something looks also in the human cells. And this might seem like a very tiny detail, but it is a real problem because this place is a landing platform for different factors. And because of human stalk being more flexible because of this missing subunit, it can bend and fit these other components better. So this can have real implications for the function of the protein. And that shows how important it is to actually look into the complex in humans and not look into some different organisms that can allow us to do important approximations, but they're still approximations. And as my last example, I would like to go back to diseases. So I mentioned Treacher-Collins syndrome, and another disease is acrofacial dysostosis. This is also craniofacial development issue. And here, throughout the structure, I marked with the balls all of the different locations of these diseases, of this mutation. So changes of one amino acid to the second one, which then causes the disease. And let us zoom out into this one particular spot. And that's how it looks when you look into the structure. So you can see this red subunit, and this in green, you have this mutated one. So it would be changed. This is the healthy situation, and it would be changed in disease. And it is an important landing platform for this another tail from another subunit, another piece. We also in the lab study RNA polymerase III, which is a related one. It makes a different type of RNA. And it so happens that this red subunit is exactly the same in RNA polymerase I and in RNA polymerase III. Yet RNA polymerase III does not have this blue piece, so it doesn't have this landing platform. And as you can see, this makes a little less interactions. So we think that even though when the person is suffering from Treacher-Collins syndrome and carries this mutation, both of these complexes will be mutated. But we think that in reality, it is issues with RNA polymerase I that will cause the disease and not RNA polymerase III, which is important for us to understand how the disease is progressing and maybe give some more predictions to children who suffer from this condition. And with that, I hope that I could convince you that there is a very, very delicate balance between having a healthy cell or an evil cancer cell. And it is quite important to maintain it for the health of the organism. And also, I hope that you could see that it is important to look into the human system directly and try to not use the approximations whenever we can, because they are useful. But if you want to think about finding drugs, perhaps it is more important to look into the system as it is. And with that, I would like to thank my supervisor, Christoph Miller, and the entire group who's been a great support, and everyone at EMBL. I was hoping that I could share with you the preprint from this work because it's coming out soon, but it's not out yet. So just watch this. You can follow me on Twitter and watch this. So it will be coming very, very soon. So then if somebody's interested, you could read the whole story. But until then, I'm happy to answer any questions. I'll also be around. So let me know. I hope that I could convince you that we actually do need basic research, at least a little bit.
Thank you so much, Agata. Good luck with final polishing your publication and your preprint. I see we have Radek Pluta with the raised hand, so I will allow him to talk now. Radek, please. Do you want to ask a question?
No, sorry, it was just by chance. But if I am already talking, then I just wanted to say it was a great presentation and also a very nice introduction for non-structural biologists to structural biology. Great work, Agata. And good luck with the paper.
Thanks a lot.
Thank you, Radek. So we have one question in the chat from Joanna Kwiatek. Hi, Agata. What does A14 little pink area play in yeast? Thank you.
So it's not doing that much, in all honesty. So it's only like 2 subunits, if I can go back. But it makes it stiffer. So so there's another bit that like another additional factor that will bind in here. So it plays the role in this binding. But also because it's there, it just kind of stiffens the whole situation. So if we compare it in yeast, it just stays like this. And in my structure in human, because we do actually have a structure with this factor, this blue bit is allowed to bend out. So this is more, it adapts. So it's more maybe specific, the binding. But yeah, that was essentially why for so long we didn't really know if there is homolog or like the version of the pink one in humans, because it's so small and so unspecific. It's like 2 helices in a tail, so it's not doing that much. But it's important anyway for stiffening.
And one more follow-up question from Joanna still. Have you checked the mutant A14 deletion?
So if you delete any of the subunits, the organism is not viable. So no. In yeast, it's actually important for some reason, or at least I think in yeast it has a growth defect only. Maybe it's not fully non-viable, but yeah, because of that, not so much. And I also don't work with yeast, honestly, so I don't.
You prefer the human model, right?
Yeah, I think it's nicer.
Okay. Well, good luck with all your studies and the publication. And I would like to thank you again for beautiful presentation with beautiful visualizations. And I think it's time for the poster session now. So please make your own coffee and come over to Gavertown to have a look on the posters. And I would like to thank all the speakers from this session and all the attendees for, well, the speakers for very interesting presentations and all attendees for cool questions. And I hope you will continue discussion on the Gavertown.